Okay, these lectures are on the spinal cord, the anatomy of the spinal cord, and as always, we're going to relate it to those functional pathways going into and out of the central nervous system. We're going to look first at the gross anatomy, looking at the whole spinal cord from the outside, as well as some relevant clinical points. Then we're going to look at sectional anatomy and all the components that you can visualize from a cross-sectional view, which is going to have critical information about what you have to know about pathways into and out of the central nervous system. And then last, we'll move on to the peripheral nervous system and talk about the spinal nerves, their structure, their distribution, and some clinical applications of that distribution. So first here with gross anatomy, we're going to look at the overall structure of the spinal cord and its location. And the first distinction I want to make here is between the spinal cord and the spinal nerves. The spinal cord is the meaty part within the vertebral canal. So the cord will contain nuclei of interneurons and motor neurons and those ascending and descending tracts. Going into and out of the spinal cord are axons of sensory and motor neurons that merge together to form the spinal nerves after exiting the vertebral foramen. The spinal cord is continuous with the medulla oblongata and so starts at the foramen magnum and continues down protected by the vertebral column as well as by the fluid-filled cavity lined by the spinal meninges, which is also continuous with the cranial meninges. From there, the spinal cord itself ends around the level of the second lumbar vertebrae, though the spinal nerves are going to continue down the column to the end of the sacrum. The spinal cord itself is surprisingly short. It's only about 1.5 feet long and 1/2 inch thick at its thickest point. The spinal regions are named after the vertebrae, although they do not line up with the vertebrae, especially in the lower regions, which I'll get to in a second. The thickness of the cervical and lumbar regions is because of the neurons associated with innervation of the limbs are located in these regions. The appendicular muscles are much more finely controlled than, say, your trunk muscles, and requires more motor neurons, and there's more sensory neurons devoted to those regions as well. Denser innervation of the limbs is also associated with those plexuses, as we will discuss later. As mentioned, the spinal cord ends around lumbar vertebrae 2. The conical tip of the most inferior portion of the spinal cord is called the conus medullaris. Below the conus medullaris, the spinal nerves continue to extend down the length of the entire vertebral column. So during early embryonic development, the length of the spinal cord is the same as the vertebral column. However, as development proceeds, the vertebral column grows faster than the spinal cord, and the spinal cord even stops growing at all at some point. So as the vertebral column lengthens, those nerves are pulled down along with the growing vertebrae. The spinal nerves are going to exit the vertebral foramen as they originally did, but their origin from the spinal cord is at a much higher region. That's why the lumbar sacral enlargement, which contains the spinal neurons controlling the lower limbs as well as the pelvic cavity organs, is way up in the vertebral column. Below the conus medullaris, the meninges that covers the spinal cord extends down into the sacral region and becomes the dural sac. So the spinal cavity containing cerebral spinal fluid is present throughout the entire vertebral column. The lumbar and sacral spinal nerves extend down from the spinal cord through the dural sac cavity and exit their respective vertebral foramen. This region that is below the conus medullaris that consists of those dangling nerves is called a cauda equina because it looks like a horse's tail. This space, which also has the cerebral spinal fluid, provides an access for that cerebral spinal fluid. In some cases, the fluid needs to be drained if there's some kind of blockage around the dural venous sinus in the brain. Remember, CSF has to be reabsorbed by the body constantly because much more cerebral spinal fluid is created than the system can hold. The second reason is that an analysis of the CSF can give a lot of information about what's going on in the brain because the CSF in that space is isolated from the rest of the body fluid. In either case, inserting a needle into the spinal cord can be very dangerous. So inserting a needle below the meat of the spinal cord minimizes the risk of poking that needle into the meat of the spinal cord. Instead, these dangling nerves can be gently pushed out of the way as the needle penetrates the subarachnoid space. And this is the same rationale for giving an epidural anesthesia below the region of L2. In this case, the needle doesn't have to go into the subarachnoid space, only into the epidural space for the anesthesia to work. But it is still safer to be below L2. Now, the epidural space is not in contact with the cerebral spinal fluid or the spinal cord tissue. And because of this, the anesthesia given in an epidural will not affect the nearby motor neurons, because those motor neuron cell bodies are located within the spinal cord. However, it does affect the nearby sensory neurons. And this is because the sensory neuron cell bodies are located outside the central nervous system. That will be covered in the next mini lecture on sectional anatomy of the spinal cord.